This video is going to have a look at how microorganisms are used to make food. In particular, it's going to have a look at how microprotein is produced, it's going to have a look at how yogurt is produced and the factors that affect it, and the advantages of using microorganisms for food production. The first thing we're going to have a look at then is microprotein. Now, microprotein is nice and simply a food that's made from fungi. So what they'll do is they'll take a fungi from mushrooms and they will squish it together and it produces microprotein, and this is used to make meat substitutes for vegetarians such as corn. So what they'll do is they'll take the actual fungus itself, which is called fusarium, and they'll grow it in fermenters. So what they'll do is they'll put the fusarium into the fermenter and use glucose syrup as a food. The fungus respires aerobically, therefore it needs lots of oxygen, which it can get from the fermenter, as you can see here. The benefits of using microprotein instead of meat is that it contains more protein and more fibre and a lot less fat. So the next section we're going to look at then is fermentation of milk to produce yogurt. So to produce yogurt then, the first thing you do is you get some milk. The second thing you do is you have to sterilise everything. This kills off any unwanted bacteria. The milk is then pasteurised, which if you remember is heated up to about 70 degrees and then cooled down to kill off any bacteria. A culture of lactobacillus bacteria is then added to the mixture and is incubated at about 40 degrees in a fermenter. As this ferments, it turns the lactose sugar in the milk into lactic acid, and that causes the milk to clot, solidify and turn into yogurt. And all that's left to do is to add flavour and colour to the yogurt so it's ready to sell. You'll also be asked to investigate the effect of different factors on yogurt making, so in particular how temperature affects yogurt production. So the first thing you do is you add 25 centimeters cubed of milk to 5 sterile boiling tubes. You then pasteurise the milk. Remember, that's by heating it up to about 70 degrees and leaving it to cool. You then add 1 centimeters cubed of the yogurt starter culture, which is the lactobacillus bacteria. Add that to each of the boiling tubes and then stir gently. The next step is to incubate the different solutions, putting them at different temperatures and then leaving them for about 5 hours. You then measure the pH of the solutions after 5 hours, and this will tell you how effective the different temperatures have been. You should find that as the yogurt's produced, the pH will start to decrease, and it should go down from about neutral pH 7 to pH 4, and that's because of the lactic acid that's been produced. So as you can see here, the ideal conditions are about 40 degrees. Anything above that and the bacteria has been killed because it's too high a temperature, and anything below that it's slightly too slow. So why use microorganisms? And there are a couple of good reasons. The first one is how quickly they grow. Bacteria reproduce every 20 minutes, which means that after a very short amount of time you've got a large amount of the bacteria grown, which when you compare this to the speed at which animals and plants grow, it has a much faster rate. Secondly, they're really easy to look after. So basically all you need is something to grow them in, which is your fermenter, and then as long as you've got food, if it needs a supply of oxygen and the right temperature, they look after themselves. It also doesn't matter what the external climate is, as long as you've got the correct temperature inside the fermenter, it doesn't matter if it's hot or cold outside. Another huge bonus is the fact that these microorganisms can use the waste products from industry as food, so it means that it doesn't go to waste. And last but not least, it's cheaper than any of the other methods.